All right, everyone, welcome back. Good afternoon. So I think we'll start off with the attendance pin. So the pin is 2747. Okay, so this week we're continuing on with our sort of look into the innate immune system. And this week we'll be focusing on what we call pattern recognition. So we'll explore that today. And in terms of the learning outcomes, these are kind of building in or building upon what you actually what we covered in the last lecture, but also kind of incorporating some new stuff. So we're sort of building on, being able to describe the properties that distinguish the innate immune system and the adaptive immune system. And we're also beginning to sort of gain some knowledge of the components of the innate immune system. So, as we talked about in the last lecture, these are the anatomical, physical, chemical, and microbial barriers. But we'll also start to introduce terms such as complement and the acute phase response. And then what we'll look at today is the innate immune cells, so the leukocytes, and we'll look at antigen recognition via pattern recognition receptors and also pathogen-associated molecular patterns. And we'll have a look at phagocytosis, inflammation, and migration. For those of you that just came, the pin is 2747. Okay, so if we start to look at the immune system, or the innate immune system, as we were looking at on Monday, we know that the innate immune system, it's ubiquitous. It's also evolutionarily older than the adaptive immune system. It likely evolved first. And as we mentioned, it's the front line of our defenses, particularly once the physical barriers have been breached. And the innate immune system does actually include the basic barrier mechanisms and also the quite sort of simplified biochemical defenses. But it also includes immune cells, so these are our leukocytes, or white blood cells, and they tend to recognize general pathogen molecules, and we term these patterns, and we'll come on to that later on. But by understanding how the innate immune system works, this will actually sort of help you understand the adaptive immune response better when we come to it in a couple of weeks' time. So as we already said, it's the first line of defense. Now, it's present at birth, and it's germline encoded. So that means that it's pretty much the same for everyone. It's not like the adaptive immune response. And, you're born with it, and it's encoded in your germline. And there are a limited number of receptors that recognize these common patterns, and these are present on the pathogens or diseased tissue. And it's important to remember that in the innate immune system, there's no memory function. So it's unlike the adaptive immune system. You get the same response each time. So there's no memory of the prior exposure. But the innate immune system helps to sort of instruct, induce, but also amplify the adaptive immune response should it be needed. Now, one of the sort of key features of the innate immune system is that it's very rapid. And so we get this immediate response from our preformed effectors. So these can be like your molecules and things like that. But then about 48 to 96 hours later, we get the induced innate immune response that is kind of dependent on the leukocytes and the white blood cells. For those of you just arriving, the pin is on the board there, 2474. So as we go along, there's a little exercise here, and you can use this as we move through the next few lectures. So basically, it's a little table comparing innate versus adaptive. So as you progress through the lectures, just try and see whether you can fill this out, building sort of, picking information from the lectures as we go along. So if we look at the innate and the adaptive immune response, so this is kind of, you'll be familiar with what we talked about on Monday. So we have our anatomical barriers, so we have our skin, our mucosa. Should that fail, we have the immediate innate immunity. So this is our antimicrobial proteins and complement. So complement is something we'll talk about next week. Should that fail, we then have the induced innate immune system. So this is our leukocytes, so our macrophages, our dendritic cells, natural killer cells. And then if that should fail, we then have the adaptive immune response. So this is when we're bringing in our T cells and our B cells, so we're getting antibody production. So let's first of all look at the leukocytes that are involved in the innate immune system. So first up, we have our granulocytes. So within this, we have our neutrophils, our eosinophils, basophils, and our mast cells. So granulocytes, they're obviously white blood cells. They tend to have these lobe nuclei, as you can see here, and they're also packed full with these cytoplasmic granules that contain cytolytic enzymes. Now, this class of leukocytes is one of the most abundant in the body. If you remember back to that blood smear that we looked at, you can see some of these different classes within that. And they really do play sort of a key role in the first line defense of the body in terms of protecting us against bacteria and various pathogens. So we run through these one at a time. First up is the neutrophils. So these are one of the most abundant classes of leukocytes in the body. They're quite short-lived, only for a matter of hours. And you can see that they're actually sort of held in reserve in the bone marrow, and then they're released in response to infection, where they'll migrate to the tissues and they'll be able to sort of phagocytose, so ingest bacteria. So they are actually dedicated phagocytes, and they provide that sort of first line of defense. And they do this by a variety of mechanisms. So we have the degranulation of the antimicrobial contents from their granules. We also have this neutrophil extracellular traps, sort of called nets, and we'll look at that in the next slide. And then they also help to initiate inflammation. This is done by releasing cytokines. So cytokines are really sort of important signaling molecules within the immune system. And if you have an infection with a pyogenic bacteria, you often get pus, and actually pus is mainly formed of dead and dying neutrophils. So these are the neutrophil nets that I mentioned. So it actually stands for neutrophil extracellular traps. So essentially what happens here, if you look in this diagram, you can see we've got a neutrophil in yellow, we've got some bacteria in red, and this blue stuff is chromatin. So in response to pathogen, what the neutrophil can do is literally vomit out its chromatin. And this forms a net that can sort of ensnare bacteria. And this chromatin will actually be decorated in a way with bactericidal proteins from the granules. So it helps to provide a physical barrier that can actually help to sort of contain and kill extracellular pathogens, whilst actually sort of minimizing the damage that can occur to the host. And they are known to sort of play an important role during sepsis, but also during preeclampsia. And we have a little video here, so hopefully, if I can get it to the right point. So this will be on the file on Canvas, if you want to look at the whole thing. And so we'll just play it here.
The nuclei of healthy neutrophils typically have several lobes, as we see here at the start of the experiment. However, after approximately 30 minutes, the nucleus in this cell, as well as many other cells, begins to disintegrate, as we can infer from the breakdown of the lobular pattern and the diffusion of blue dye throughout its cytoplasm. After an additional 15 minutes, the cell extrudes its contents into the extracellular space, as shown by the green arrows. As the experiment continues, additional neutrophils undergo netosis until about half have generated extracellular nets.
So, yeah, if you want to watch that in more time, you can find that video on the PowerPoint on Canvas. But essentially, you see there the nuclei of the cells breaking down, the chromatin being extruded, and that will actually sort of ensnare any of those pathogens. The next granulocytes to look at are the eosinophils. So these will have quite a characteristic staining. And you probably see that on the blood smear from the last lecture. But they stain brightly, sort of a pink bright staining when you use this HE staining. They also have this bilobed nucleus, and they play a really beneficial role in parasitic infections. But they also play a damaging part in the allergic response, because you can imagine that in the sort of the Western world, we don't really encounter parasitic infections that much. So we sort of get a little bit of an immune response that's a little bit kind of skewed in a way, and this is where you can have issues with allergy. So obviously, because we don't encounter parasites that often, we tend to sort of have more instances of allergy, and these cells do play an important response with that. They're also recruited to the tissues at the sites of inflammation. They play an important role in releasing these antimicrobial factors, They also have the potential to present antigen to T cells, so that's something we'll look at later on in the module. And they also help to initiate inflammation, again by releasing these cytokines, which are the important signaling molecules of the immune system. If we look at basophils, you can see that again they have this quite distinct sort of cell size and shape, and they're packed full of these granulars, so they're really sort of highly granular. They form less than 1% of all leukocytes, but again, they also play an important response to parasites and allergy. Again, they're recruited to sites of inflammation, but in this instance, they actually become activated by the cross-linking of surface-bound antibody. And again, that's something we will look at in the future. And once they're activated, they degranulate, and they help to sort of release these inflammatory mediators. And then we have our mast cells, which are one of the first cells to play an important role in allergy. Again, they would normally be involved in a sort of parasitic response. They're tissue resident, so they're found in the skin, they line the blood vessels, as well as being present in the mucosal tissue. And these also get activated by the cross-linking of surface-bound antibodies. So you can see here we've got a mast cell, we've got cross-linking of surface-bound antibody, recognizing an antigen, and we're getting degranulation. So if you suffer from allergy, you'll probably be familiar with histamine because you might take antihistamines. So when the mast cell becomes activated by cross-linking, it degranulates, it releases histamine, and you get effects such as you get vasoactive effects, but you also get smooth muscle contraction, because obviously these responses were kind of designed to eliminate or get rid of a pathogen or a parasite, from our gut. But obviously in allergy, that's a bit misdirected, so you get all the symptoms of allergy, basically. They also play an important role in releasing inflammatory cytokines, and this helps to promote a Th2 adaptive response. So again, that is something we will look at in the future weeks, but the Th2 response plays an important role in allergy as well as parasitic infections. Now, I just wanted to refresh your memory on phagocytosis. So in this diagram here, we have a macrophage. We also have a bacteria that's coming along. It's being recognized by the macrophage, and it's being engulfed. And then what will happen is we'll get the formation of a phagosome, so the bacteria is internalized, and it will then be degraded as this phagosome sort of turns into a phagolysosome. So this is one way that the macrophage can actually engulf and destroy pathogens, and this is via phagocytosis. So we look at some of the phagocytic cells in our innate immune system. We have the monocytes, macrophages, and the dendritic cells. Now, monocytes are actually the precursor to tissue macrophages. And here we have the macrophages. So macrophages are really specialized phagocytes. They're specialized for phagocytosis, and they're able to present antigen that they encounter to the T cells of the adaptive immune response. And then the third cell that you will remember hopefully from Monday is the dendritic cell. And this is one of the most important antigen-presenting cells, and it plays a really key role in presenting antigen from the innate immune system to the adaptive immune system, specifically through T cells. And if you have a look, you'll see that it's got these long processes, these long dendrites, and it's also packed full of lysozyme as well. So if we focus initially on monocytes and macrophages, so this would be a monocyte at the top, and then this would be a macrophage. So as I mentioned, the monocyte is the precursor to macrophages, but it's also a precursor to some classes of dendritic cells. Monocytes will be recruited to the tissues at sites of inflammation, and they can then differentiate into macrophages. And as we already said, macrophages are professional phagocytes, so what they'll do is they'll recognize pathogens or foreign particles, and they will engulf it, so they'll phagocytose it, and you can see that in the diagram here. We've got a macrophage phagocytosing and engulfing some bacteria. In this case, it's anthrax. And what they also do is they play a really important role in releasing antimicrobial factors, but also releasing pro-inflammatory cytokines, so they build on that sort of inflammatory response. And again, they have this antigen-presenting capacity. Now, if we compare neutrophils to macrophages, so we look at them, first of all, in terms of the life cycle. Neutrophils increase in numbers during infection. They migrate into the inflamed tissue, but they don't last that long, so they die quite quickly after migrating into the tissue. If we compare that to macrophages, macrophages are actually produced at a steady level. They migrate into the tissue in the absence of inflammation, but they can actually survive for much longer. So they can survive for quite a few years, even after they've encountered pathogen. In terms of the mechanisms of killing pathogen, both neutrophils and macrophages can perform phagocytosis, and they can also kill using these toxic molecules. But one difference between them is netosis. So neutrophils can do this netosis, so extruding this chromatin, but macrophages have no netosis. And when it comes to the communication with other parts of the immune system, neutrophils don't have any role sort of in instructing the adaptive immune response, where macrophages can actually present antigen to the adaptive immune response. So they kind of stimulate the adaptive immune response via antigen presentation. So in terms of actually sort of the adaptive immune response, so this is a slide that we looked at in the last lecture. So we've got adaptive cells, adaptive immune cells, we've got our T cells, our B cells, and our plasma cells. And if you remember, we were saying that the adaptive immune response can only be switched on through the actions of the innate immune system. So the innate immune system kind of, is bringing in the big guns of the adaptive immune system. And we mentioned that the most powerful cell that actually sort of communicates between the innate and the adaptive immune response is the dendritic cell. And this, again, is a professional antigen-presenting cell. Now, we've already said that the macrophages, the monocytes, the neutrophils, and the dendritic cells are all professional phagocytes, and they're all very effective at phagocytosing bacteria, parasites, or cell debris. Now, even though they're very efficient, the actual outcomes from this phagocytosis can be quite different when it comes to dendritic cells. So the macrophages, monocytes, and neutrophils will essentially scavenge, but then they'll phagocytose particles, but they'll destroy it. And that's actually quite an important role in the innate immune system. But in contrast, the dendritic cells tend to preserve that useful information because their main role is to present it to the adaptive immune response. So in that sense, they're quite good at preserving the material that they actually ingest. And then they will present that to the T cells. So if we look back at inflammation, and this is a diagram that we came across in the last lecture. So just to recap what inflammation is, it can be the local or systemic response to either injury or infection. So, we're sensing either damage or we're sensing a pathogen. It can also be systemic. So if you think about sepsis, that's actually a systemic inflammation. But what we're getting is we're getting this recruitment of immune cells and molecules whose main job is to eliminate that infection, but also to initiate repair. And it's coordinated by the leukocytes that we looked at, but it also, there's an important role in leukocyte traffic. So by traffic, we mean the migration of the leukocytes into that inflamed tissue. Inflammation will normally be acute, so by that we mean short-term, because obviously if you think about inflammation, it has the potential to cause damage to your surrounding tissue. So you really want to turn it off when it's not needed in order to minimize the amount of damage that actually could potentially occur to your body tissues. But sometimes inflammation can become dysregulated, and we call that chronic inflammation. So, if it's dysregulated, if it's recurrent or it's persistent, you get this chronic inflammation, and obviously that's very problematic because it can cause damage to the surrounding tissues, and we really want to avoid chronic inflammation. So this figure is just summarizing the different stages of infection and how we can set it into sort of different steps, basically. So here we have the normal situation. We may have some pathogens associated with the epithelium, but if our physical barrier gets damaged, the bacteria can sort of enter the body, and it will be detected by our resident innate immune cells, and we'll have sort of production of antimicrobial proteins. We'll also have complement there. But if the innate immune system can't control the infection, we'll start to get this localized infection of the tissues. And what happens then is we'll have our dendritic cell here that will be picking up parts of the pathogen, and it will start to migrate towards the lymph nodes. And once it migrates to the lymph nodes, it will encounter T cells, and it will instruct the adaptive immune response. And then eventually we get sort of full engagement of the adaptive immune response. We'll get production of antibodies, we'll get B cells and T cells, and we'll get this influx into the sort of inflamed tissue. But essentially, if the innate immune system is unable to actually tackle the infection itself, we'll then recruit and involve the adaptive immune response. So how are the innate immune cells activated? How do we control this whole thing? Well, first off, we have these inflammatory inducers. So an inflammatory inducer, it can be a chemical structure that indicates the presence of an invading microbe. So this can be on the surface of a pathogen, but it can also be cellular damage that's perhaps produced by a pathogen. And these are known as pathogen-associated molecular patterns. So remember at the beginning of the lecture we talked about patterns. So the pathogen-associated molecular patterns will be found on a pathogen, and it's abbreviated to PAMPs. But we also have damage-associated molecular patterns, which are known as DAMs. So these kind of indicate the presence of a pathogen or the presence of damage. We then have our sensor cells of the innate immune system. So these will be our macrophages, our neutrophils, our dendritic cells. And these will contain, or these will have, these innate recognition receptors on their surface, which are called pattern recognition receptors, or PRRs. And the job of the pattern recognition receptor is to recognize either the PAMP or the DAMP, okay? And upon sort of recognition of these inflammatory inducers, the innate immune system, or the innate immune cells, can actually reduce a variety of inflammatory mediators. So these can be our cytokines, which are our cell signaling molecules. And these essentially they can play a direct role in the defense, or they can help to kind of further propagate our immune response. And so as already mentioned, these mediators include cytokines, and they can have basically a function to act on epithelial cells to actually induce the production of antimicrobial proteins, and that will help in our immune response and hopefully fight off the pathogen, but they can also help to amplify that immune response. And we'll kind of go through cytokines throughout the module as well. So just to kind of recap that, and this is actually a really kind of fundamental concept in terms of understanding the immune response, but the immune system plays a really key role in determining self and non-self. So when we say self, we mean our own body tissues, and non-self would obviously be pathogens. And basically the distinction between self and non-self, it can be done at the level of the innate immune system, but also at the level of the adaptive immune response. But these pattern recognition receptors, so the PRRs, play a really key role in discriminating self between non-self. So they're able to recognize the pathogen-associated molecular patterns on a pathogen, which is obviously non-self, but then they're also able to recognize the damage-associated molecular patterns that can be associated with perhaps infection or transformation of a cell, so malignancy, and that's how the innate immune system can play a role in actually detecting a malignant cell or a transformed cell. So that's really a key process that you need to kind of, remember and get your heads wrapped around. So if we look a little bit more detail at the pattern recognition receptors. So as we mentioned, these play a really key role in distinguishing between our own body cells, our own healthy cells, and perhaps a transformed or infected cell or a pathogen. But in the diagram here, we have our macrophage, we have a bacterium, and we can see that the pattern recognition receptors are recognizing these general patterns on the surface of the bacteria. And that's how we can actually identify, or our innate immune cell can actually identify these non-self pathogens. But also the pattern recognition receptor can recognize changes on the surface of our own body cells. So here we have an epithelial cell, and it's been infected with a virus. This causes changes on the cell's surface, and this can be detected by the cells of our immune system. And likewise, if this could equally be a transformed cell or a malignant cell. So in that respect, it's the PIRs that play this key role in being able to determine self from non-self. So we look at some examples of the PAMPs and the DAMPs. So this table just kind of summarizes that. So in terms of the PAMPs, we can have peptidoglycan, which can be found on the surface of gram-positive bacteria. We can have lipopolysaccharide, which will be present on the surface of gram-negative bacteria. We can also have flagellin. In terms of viruses, we might have double-stranded RNA. And then for the damage-associated molecular patterns, we have DNA that's outside of a nucleus, serum amyloid A, also ATP or uric acid. There are other examples of damage-associated molecular patterns. Basically, something where it shouldn't be is also a good example of basically damage, so that will serve as a damage-associated molecular pattern. And these are all recognized, so PAMPs and DAMPs are all recognized by your pattern recognition receptors. Now, you might recognize the name Janeway. So the core textbook, Janeway's Immunobiology, it was actually first written by Charlie Janeway. And he's kind of one of the sort of like leading immunologists of his day. But he proposed this pattern recognition hypothesis. And basically, he was predicting that we're getting the activation of the adaptive immune response being controlled by the innate immune system. And this pattern recognition hypothesis is basically this. So classes of germs will carry molecular patterns, and these are either anchored on the surfaces or secreted from the insides. But all multicellular organisms will have pattern recognition receptors that can signal, only in vertebrates, the adaptive immune system. But of course, the microbial molecules that carry the patterns would have to be critical to the pathogen in some way. Otherwise, it would have evolved a way to do without those molecular patterns a long time ago. So, he played a key role actually in kind of promoting this hypothesis. But it kind of all stems from his work. And actually, like I said, he was the main author for this textbook, which is actually a very, very good textbook. In terms of the pattern recognition receptor, they can fall into several classes. So we have our toll-like receptors, or our TLRs. We have our C-type lectin receptors, our RIG1-like receptors, but also our NOD-like receptors. And we'll mainly focus today on the TLRs and the NOD-like receptors as well. But essentially, the function of these is they will activate these signaling pathways, and the ultimate result is that we'll get gene transcription that will result in the formation of proteins. So these can be cytokines, and then these cytokines can go on to contribute to the inflammatory response. So if we focus now on the TLRs and the NLRs, so the toll-like receptors are transmembrane pattern recognition receptors, so you can see them on the surface here. And then the NOD-like receptors are actually intracellular ones. And we'll look at these in more detail in the next coming slides. But the NOD-like receptor is intracellular, and it will play a key role in detecting those DAMPs and PAMPs that are within the cell itself. We also have some other receptors. So we have the mannose receptor, the glucan receptor, and the scavenger receptor. And you can see that on the surface here. And these all play a key role in actually helping to sort of detect and bind to the carbohydrates that we might find on the surface of a pathogen. But it's worth bearing in mind that the innate immune cells will actually sort of express a variety of these different receptors that will enable us to actually enable the cells to detect pathogens. So we first look at the TLRs. So these are highly evolutionary conserved, and they actually sort of form, an ancient pattern recognition system. Originally, they were discovered back in 1985 in Drosophila. And we can see them on the surface of this cell here. So they can be on the cell surface. They can also be found in endosomes. But essentially, their main function is to detect either the PAMPs or the DAMPs. So an example of this would be detecting lipopolysaccharide on a pathogen. And if the toll-like receptor is on the cell surface, it helps to basically transmit that signal across the cell membrane to the cell interior, where we can then get a signaling cascade which will result in effector functions. So this can be the cytokine production to propagate that signaling response, or chemokines. So chemokines are important in sort of migration of cells, and we'll touch on that in the coming lectures as well. And if we look at the different varieties of toll-like receptors, you can see in this diagram here that they can exist as either homodimers or heterodimers. They have different locations in the cell, so they can be on the cell surface. They can be found, as I mentioned before, on the endosomes. They have different distributions across the cell, but they also have a capacity for different ligands. So you can see here at the top that they sort of are specialized in binding to different things. But essentially, the main outcome of this is that they will all activate NF-kappa B. So NF-kappa B is a transcription factor, and it's one of the main transcription factors in the inflammatory response. But activation of NF-kappa B will lead to the production of inflammatory cytokines, chemokines, as well as antimicrobial and antiviral factors. So even though we have a whole variety of toll-like receptors, we're basically getting that activation of the NF-kappa B transcription factor. If we switch now to look at our NOD-like receptors, so when it comes to that, we've got about 23 plus cytosolic pattern recognition receptors that can actually detect this intracellular PAMPs or DAMPs. They're present in macrophages, dendritic cells, but also epithelial cells. And again, like the TLRs, they activate the NF-kappa B transcription factor. But the NOD-like receptors actually play a key role in activating what's known as the inflammasome. And so we'll go through that on the next slide. But essentially, it's what you want to take home from this is that we still have this similar downstream signaling which is being mediated via the activation of NF-kappa B. So this diagram is just summarizing the inflammasome. So what we're doing here is we're looking at, we've got a macrophage here, and then we've got the extracellular environment there. So we look from basically where I've got this start arrow. If we have damage to the cell, we'll have changes in the levels of potassium, but also ATP. And this will act as a DAMP, so a damage-associated molecular pattern. And you can see that within the macrophage, we've got our NOD-like receptor here. So in this instance, this one is known as NALP3. And we also have an adaptor protein and procaspase 1. But in the presence of the DAMP, so the NOD-like receptor will actually detect, let's say, the ATP changes in the ATP levels or the potassium levels. We'll get this oligomerization between NALP3, the adaptor protein, and procaspase 1. And we get this formation of this inflammasome here. Now, once we've got the formation of the inflammasome, we get sort of release of active caspase 1. And this active caspase 1 will actually be able to cleave pro-IL-1 beta. So pro-IL-1 beta is inactive. Once it's been cleaved by caspase 1, we'll get release of IL-1 beta. And IL-1 beta can be secreted. IL-1 beta is a cytokine. And what it does is it has a really sort of broad function in the body, and it plays a really key role in regulating fever. Now, you can imagine that obviously fever is something that needs to be regulated, and therefore we need to ensure that IL-1 beta is released appropriately and only when it's absolutely necessary for the body to do so. And this helps to mediate what we call the acute phase response. And the acute phase response is essentially a systemic reaction to disturbances in its homeostasis that's been induced by either tissue damage or infection. And so this is helping to mediate fever and basically sort of helping in that anti-pathogen response. An example of an auto-inflammatory condition would be gout. And I've put here, it's really important that you don't confuse auto-inflammatory with autoimmune. So auto-inflammatory would be dealing with aspects of the innate immune system, whereas autoimmune would be dealing with aspects of the adaptive immune response. So they're actually controlled by different aspects of the immune system. Now, gout is actually a type of arthritis, and it occurs in response to the buildup of uric acid in the joints. So you can have that in the joints of the feet or the hands, the fingers, for example. Now, uric acid is actually a natural byproduct, that's formed by the breakdown of purines that are found in meat and seafood. And the uric acid actually serves as a DAMP, so a damage-associated molecular pattern, and that uric acid will activate the inflammasome. And obviously you can imagine that once we've got activation of the inflammasome, we're getting activation of IL-1, and we're getting recruitment of neutrophils, and this is all being recruited to the joint, and we're getting this basically auto-inflammatory response in the joint. So we're getting this deposition of uric acid, we're getting activation of the inflammasome, and we're getting this this influx of neutrophils and macrophages as well. Okay, so it's an auto-inflammatory condition that's mediated by the innate immune response. So we've got a little quick quiz now. So it's just 6 questions. So if you go on Poll Everywhere and have a go at that, and that will just sort of test your knowledge of the last couple of lectures. And I'll just give you a few minutes to do that. Okay, it looks like you're beginning to get there. Just give you a few more. All right. I think we'll start to run through it now. So the first question, a term that's used to describe all white blood cells, it is, of course, leukocytes, and the majority of you got that. So that's looking good. Which of the following forms an important link between the innate and the adaptive immune response? Dendritic cells, so again, the majority of you got that one. Which of the following are characteristics of the innate immune system? Inflammation and a fast response, so remember it's that immediate aspect of the innate immune system that's actually one of its benefits. In terms of recognition of pathogen and specificity, these are the two sort of key features of the adaptive immune response that we'll talk about later on. Which of the following events doesn't occur during inflammation? It is, of course, constriction of blood vessels, because that's one of the things that you wouldn't want in inflammation. Inflammation is actually the opposite. And all of these are features of inflammation. So we will talk in the coming weeks about cytokines and chemokines. So chemokines play that important role in basically migrating cells, so the migration of cells. Which of the following is a peripheral lymphoid organ? So the spleen and the appendix, the bone marrow and the thymus, if you remember from Monday, are actually the primary lymphoid organs. And what is common to macrophages and neutrophils in a healthy individual? It is, of course, phagocytosis. Okay, so that looks really good. If we just flick back to here. So just to summarize what we've been talking about sort of today, and also basically this week. But we have our basic barrier mechanisms, so these can be biochemical but microbiological defenses, and these actually form the front line of the innate immune response. And that the innate immune response actually does depend on being able to recognize what we call these general pathogen-associated molecular patterns, and these are recognized by PIRs, and the PIRs can also recognize damage-associated molecular patterns that will signal, which will signal damage that can be generated via a pathogen, a virally infected cell, or transformation in terms of cancer. But basically, the outcome of triggering the PIR on the innate immune system can be varied, but it will include activation of complement, which we will discuss next week. Also, inflammation and the acute phase responses, which we'll go into more detail. Phagocytosis, again, we'll go into more detail on that. But also triggering the activation of the NF-kappa B transcription factor and helping to initiate, essentially the adaptive immune response. But by understanding the innate immune response and these early steps, you'll be able to get a better understanding of the adaptive immune system. And then I put here a little sort of exercise slide. Essentially, it's recapping everything that we've covered this week. So you can do this by yourself or amongst your friends as well, but just see whether you can discuss the following questions and just basically, whether you can answer and just write out a list for all of these things. And then next week, what we'll do is we'll start to look at phagocytosis, inflammation, complement, but also exploring those innate infector mechanisms. And in terms of reading, these are the sort of chapters that are good from Kubi and also Janeway Immunobiology. But that's it for today, and I'll see you guys on Monday morning. Okay?